Hello. I had the privilege of training my postdoc at the University of Pennsylvania in the laboratory of Carl June, and during that time, our group was the first to present clinical data on anti-CD19 CAR T cells in chronic lymphocytic leukemia, and then later in acute lymphoblastic leukemia. And the impact of these therapies, as Stacy mentioned, is seen in summary in the pictures of the patients. So this was the first pediatric patient treated. Her name is Emily Whitehead, and this is her before treatment, and this was her last month, 4 years in remission. And some of these patients have become international superstars. Emily stood next to President Obama as he signed the Precision Medicine Initiative in 2015 because she's a face of personalized and precision medicine. And this is patient number 15, Nicholas Wilkins, who met the Pope last month and at the Vatican on a conference on emerging cell therapies. So the CAR T cell treatment starts with leukapheresis of these cancer patients. Their T cells are then activated with antibody-coated beads and in our case, transduced with a lentivirus to express the chimeric antigen receptor, and those cells are then expanded in culture, the beads are removed, and then returned back into a lymphodepleted patient. And so the molecule that we use, as Stacey mentioned before, provides antigen specificity or antibody-driven specificity to T cells against a particular cancer antigen using a chimeric molecule that's an artificial T cell receptor. And so you get T cell activation and co-stimulation that leads to survival and persistence in some cases. And so our model is we use a patient polyclonal T cell population, transduce them with a lentivirus to express the chimeric antigen receptor. And that receptor now gives the T cell bispecificity, one through its endogenous T cell receptor, and then through the CAR construct for a tumor molecule, such as CD19, and then those T cells can then recognize the tumor and kill it. And so this is the first patient treated with CLL, and this is the patient's cells 1 day before T cell infusion and 1 month after, and you see there's a complete eradication of the CD19 positive cells. So these CAR T cells target all of the target cells in less than a month, and in some patients it's been as few as 10 days, and these are patients who have gone through multiple rounds of chemotherapy and radiation and are relapsed or refractory, have no other treatment options. And this works very well for patients with leukemia because humans can live without B cells. But this does not work for solid tumors, and you need targets that can differentiate between normal tissue and solid and cancer. And so my approach has been to exploit a mechanism of to exploit differential glycosylation in cancer. So it's known that cancer cells have a defect of O-glycosylation profile that leads to hypoglycosylation of olein glycoproteins, such as the presentation of the TN antigen or SIL-TN antigen, while normal tissue is fully glycosylated. And so we collaborated with investigators at the University of Copenhagen, and we demonstrated that in normal MUC1 positive tissue, such as kidney, normal MUC1 is expressed at the cell surface, such as in the distal tube, in the lumen of the distal tubules. But the cancer-specific, I'm sorry you can't see this, but the cancer-specific version is only expressed intracellular, and this is intermediate glycoprotein in terms of full glycosylation. However, in cancer cells, this TN-MUC1 is expressed brightly at the cellular surface. So we developed a chimeric antigen receptor using an antibody that recognizes the cancer-specific version of MUC1. And we can show that when we include cells together in the same well that have either the TN expression or full glycosylation, that our CAR T cells will target only those that have defective glycosylation. So those CAR T cells can differentiate hypoglycosylated or cancer-specific cells from normally glycosylated cells in the same environment, demonstrating safety. And so we've shown that those CAR T cells can now recognize a wide variety of cancer cell lines. And in a model of metastatic pancreatic cancer, we show 100% survival of mice with this pancreatic cancer that were treated with this new CAR. And so this work is published on Tuesday in Immunity, and I'm lucky we've gotten press releases from Cell, Science, and Forbes, and I'm very excited about this work. My lab will focus on developing new classes of CAR T cells targeting glycoantigens. Thank you.
I have a question. So have you tried to put lectins and just, direct lectins against instead of having with the antibodies? You do this with lectin, and then what happens with the, I guess, intercellular or the, actually in blood concentration of different heptanes, the glucose? Would they knock off these interactions?
I'm sorry, I missed the second part of your question. But the first part, so in our experience, lectins are not very cancer specific because they only target the terminal glycans. So there's the potential for off-tumor, on-target toxicity. And the second part of your question? Okay.
So related to Julien's question, because you and Stacy do similar work, have you guys used this program to collaborate and think very strategically and maybe use her computational biology methods to make your science even better?
We've had communication on this, yes. Thank you.